Dear students, Module 15. Duplication with suitable examples. The main objectives are to know about the chromosomal abnormalities and their consequences, to explain about the duplication along with their classifications, to understand the role of duplication in the evolution of organism, and to know about some of the genetic disorders caused due to duplications, to explain the significance of duplication along with their origin. Introduction. In nature, the naturally occurring operations like mutations and crossing over provide one way to alter the genomic content. For example, the mutation will alter the chromosomal string by changing one nucleotide base of that string. When this changed DNA gets translated into protein inside the cell, the mutation may lead either to the abnormal functioning protein or no production of protein. This variant of the protein may then affect the structure and behavior of the organism in some advantageous or disadvantageous way. In some cases, if the change is beneficial, natural selection will tend to retain those changes. In most cases, the random mutation can be disadvantageous and natural selection will tend to cause the extinction of the mutant. Chromosomal rearrangements can also occur by other naturally occurring genetic events such as gene duplications and gene deletions. These are some structural chromosomal abnormalities results from the breakage and incorrect rejoining of chromosomal segments. Gene duplications are unpredictable and rare events in the evolution of genomic sequences. In gene duplication, the part of the genomic content gets doubled to have one more copy. This type of structural abnormality comes under the unbalanced rearrangements results from the addition of missing of genetic material. When the gene is duplicated, there may not be an immediate change in the production of protein inside the cell. This can become as two identical ways of the same manufacturing protein. Over some time, any genetic event such as mutation or crossing over may change one of the two identical genes, but the remaining copy of the gene can continue to manufacture of the original protein. In some cases, significant effects of additional genetic functions can result in phenotypic changes. In this episode, the following aspects are emphasized: duplication and its classification, causes for the origin of duplications, disorders caused due to duplication in human, and significance of duplication. A. Duplication and its classification. Duplication is one of the structural chromosomal anomalies which sometimes produce an extra copy of the chromosome region. Bridges first coined the term duplication in 1990. The first duplication was described in the fruit fly that is Drosophila melanogaster. A small piece of a chromosome can contain many different genes, and if the extra gene is present in duplication, may cause those genes not to function correctly. This can lead to errors in the development of an infant due to the extra instructions. In the case of a haploid organism, which usually has one chromosome set or haploid set, the duplication can be easily recognized by the presence of two sets of chromosomal regions. Duplication is mainly classified into interchromosomal duplication and intrachromosomal duplication. This classification is based on the occurrence of duplication on the chromosome. Interchromosomal duplication is where the duplicated segment of the chromosome is present in another chromosome in the genome. In intrachromosomal duplication, the duplicated segment remains in the same chromosome. The duplicated region may find at different locations. It can be in the same arm, but removed from the original segment, or it can be in the same arm and next to the original segment. Duplicated segment locating just next to the original segment is referred to as tandem duplication. The gene in the duplicated segment is similarly to the sequence of genes in the original segment of a chromosome. Tandem duplication further shows direct tandem and reverse tandem duplications. In direct tandem, the gene order in the duplicated region is similar to as the gene order in the original region. In the case of reverse tandem, the order of the gene in the duplicated segment is inverted. The interchromosomal duplication is further shown two subtypes, where the duplicated segment of a chromosome can incorporate into a non-homologous chromosome called as translocated duplication. Another subtype where the duplicated segment is included away from the corresponding segment on the same chromosome is called as displaced duplication. In some cases, the duplicated segment is present as a separate chromosome. The possibility of survival is less until it gets a centromere, formation of duplication loop. The duplicated segment can form a loop during pachytene in duplication heterozygotes. If the duplication heterozygote is further undergone unequal crossing over, it can lead to the formation of further duplication of the concerned segment. B. Causes for the origin of the duplication. In a diploid organism, the chromosome set containing duplication will present together with a standard chromosome set. In those cases, cell of such an organism will have 3 copies of the chromosome regions. In such cases, the tandem repeat heterozygotes during meiotic prophase show the formation of the loop, which represents the unpaired extra region. The origin of this duplication may take place in the following ways. change in the primary structure of chromosomes. If a chromosome break by the external factor such as radiations, chemicals, etc., the broken segment of a chromosome may be inserted into its homolog or non-homologous chromosome. In 1950, Barbara McClintock described the dissociation activator system in maize. This was a very remarkable case of genetically governed production of aberrations. Both DS and AC are capable of transposition to any chromosome or within the same chromosome. During the transposition of DS, a break occurs at the location where it was inserted. Hence, the cytological effects produced by these systems create various kinds of chromosomal aberrations such as deficiencies, duplications, and etc. Unequal crossing over, the improper pairing of chromosomes, and crossing over processes or deviation that occur during the meiotic stages. They can mainly occur in specific cases in heterochromatic regions. This kind of pairing is termed as a heterochromatic fusion or non-specific pairing. This can lead to unequal crossing over. It was observed in the bar I locus of Drosophila melanogaster. The unequal crossing over in the X chromosome of Drosophila resulted in a duplication of 16A region. This duplication produces the bar I phenotype. If two X chromosomes with bar I locus are undergoing unequal crossing over, they result in one X chromosome with duplicated 16A region and another X chromosome remain deficient for the 16S locus. If bar I homozygote is undergoing further unequal crossing over, it results in double bar, means ultra bar I locus, 316S locus in one chromosome and normal locus in another chromosome. Crossing over within inversion can produce the chromosome showing the translocation heterozygotes will produce deficiency, duplication as well as the deficiency duplication gametes. These formed gametes will remain sterile. In 1965, Hegberg produced duplication in barley using translocations. Disorders caused due to duplication in human. Duplication in humans may result in some of the diseases that show facial, dysmorphic and congenital anomalies as main features. Some of the human disorders include Charcot-Marie tooth disease and Pillsbury-Killian mosaic syndrome. 1. Charcot-Marie tooth disease is also called as CMT, is the most common inherited neurological disorder affecting approximately 1 in 2,500 people in the United States. This disease was named for the three physicians in 1886, namely Harvard Henry Tooth in Cambridge, UK, Jean Martin Charcot and Pierre Marie in Paris, France. CMT is known as hereditary motor and sensory neuropathy, or peroneal muscular atrophy comprises a group of disorders that affect peripheral nerves. Diseases that show the defect in the peripheral nerves are referred peripheral neuropathies. Motor nerves control the voluntary muscle activity such as walking, speaking, swallowing and breathing. Based on electrophysiological and pathological studies, CMT has been divided into two large distinct groups. CMT type 1, the demyelinating type, that shows mild to harsh reduction in motor nerve conduction velocity, absent muscle stretch reflexes, and the formation of onion bulb appears in nerve biopsy. CMT type 2, the neuronal form, shows a normal or moderate reduction in motor NCVs and decreased amplitude, normal muscle stretch reflexes and no hypertrophic characteristics on nerve biopsy. The gene mutation in CMT disease are usually inherited. Although different proteins are abnormal in various forms of CMT disease, all of the mutations affect the normal function of the peripheral nerves. Most cases of CMT are associated with a 1.5 MB tandem duplication in chromosome 17, that is 17p11.2 p12, that encompasses the PMP22 gene. CMT1A is the first autosomal dominant disease associated with gene dosage effects due to inherited DNA arrangements. There is no mutant gene, but instead the disease phenotype results from having 3 copies of a normal gene. The degeneration of motor nerves resulted in muscle weakness and atrophy in the extremities. In some cases, the degeneration of sensory nerve occurs in a reduced ability to feel heat, cold and pain. Significant clinical symptoms of CMT disease are a combination of the motor and sensory neuron type, which shows the sensory motor neuropathy. The symptoms are length dependent, which can be partial paralysis or result in muscle atrophy, which leads to loss of reflexes, but several patients will keep the reflexes in the axonal type of CMT disease. Symptoms of the sensory dysfunction frequently appeared in almost 70% patients and include the failure of vibration, joint sensation succeeded by the loss of pain and temperature sensation in hand and foot. The onset of symptoms is most often in adolescence or early adulthood, but some individuals develop symptoms in mid-adulthood. The severity of symptoms varies significantly among individuals and even among family members with the disease. 2. Pelletier-Killian mosaic syndrome. It is a developmental disorder that affects any part of the body. This syndrome appears to be a rare condition. It is usually caused by the presence of an abnormal extra chromosome called an isochromosome 12p. Isochromosome is a chromosome with two identical arms. This chromosome can have either two p arms or two q arms. In people with this syndrome, will have two usual copies of the chromosome 12p and an isochromosome 12p, which makes 4 copies of all the genes which are present on chromosome 12p arm. Therefore, the extra genes from the isochromosome disrupt the normal course of development, causing the characteristic feature of this disorder. Pallister-Killian mosaic syndrome is not inherited. The chromosomal change responsible for this disorder usually occurs as a random event during the formation of reproductive cells. The syndrome is characterized by extremely weak muscle tone in infancy and early childhood, intellectual disability, distinctive facial features, areas of unusual skin coloring or pigmentation, and other congenital disabilities. Additional features of this condition include hearing loss, vision impairment, seizures, extra nipples, genital abnormalities, and heart defects. The severe cases involve congenital disability that are life-threatening in early infancy. D. Significance of duplication. Duplication can also cause the position effect. The position effects can be stable or not. Example, the Barré phenotype of the Drosophila. The variegated type position effects are confined to the gene present in the heterozygote state. It is the result of partial repression through heterochromatinization when the functional allele of a gene is brought close to heterochromatin. The standard wild type allele expresses like a mutant allele due to the heterochromatinization. A duplication of a concerned gene increases the activity of a specific enzyme. Duplications are believed to have played an essential role in evolution. In Drosophila, the pollinating chromosomes from salivary glands can be analyzed accurately. Numerous duplicated segments have been identified. Many duplicated loci are investigated in plants also. Duplications also give rise to new gene functions. Duplications are used to study the chromosome behavior during meiosis, namely chromosome pairing, crossing over, and their consequences. It can offer several possibilities in plant breeding. Duplication can be used to increase the dose of specific desirable genes for increasing pest or disease resistance and enzymatic activity. The activity of enzyme alpha amylase in barley grains is significantly increased due to duplication in the short arm of chromosome 6. Duplication can be used to study the dosage effect of the nucleolar organizer. By using the duplication, breeding, true breeding, heterosis may be established in self-fertilized crops. In several regions of the genome showed gene duplication which affect the variety of traits and diseases in humans. To conclude, the structural chromosomal aberrations such as duplications have a very important role in the evolution and helps in much genetic analysis. Thank you.